Hi.
Hi, Aisha. How are you?
I'm good. What's your name?
My name is Taramina Lapori.
Oh. What field did you do your major in?
I studied biology. I have a Bachelor of Science degree.
Oh, I see. I've heard you've made drugs. How was your experience?
That's an interesting way to put it. Yes, I did make legal drugs, medications, and my experience was very interesting. I worked for a really big pharmaceutical company or drug company, and this company makes a lot of different things, everything from chapstick to Robitussin to prescription medications. And what I did in that particular company is I working on medications that were going out for clinical testing. And do you know what a clinical trial is? Well, a clinical trial is when a medicine is put out for a trial or a test. So it hasn't gone out to everybody in the entire world yet, but it's gone out to test a few select populations to see how effective it is.
That's interesting.
Yeah.
Have you had experiences of extracting DNA before?
Yes, I have. Now, I didn't extract DNA when I worked in the pharmaceutical industry, but I did in college as a biology major. I worked for a little while in what's called a molecular phylogeny lab. Do you know phylogeny? Well, phylogeny is when you look at how animals are related through something called a phylogenetic tree. You might have seen pictures of phylogenetic trees, and molecular phylogeny looks at how animals are related based on their genes.
What else can be DNA extracted from other than strawberries?
Well, interesting you should ask that. Other than strawberries, we can extract DNA from our own cells. If you take a swab of your cheek cells and even use simple everyday items, you can mix a concoction together that will extract DNA painfully, harmlessly, and interestingly. And when I was in college, my job was to extract DNA from a type of parasitic insect called a scale insect, and they were infecting pine trees back in Massachusetts. So we were looking at how they were related to each other, species to species, and we had to grind up a bunch of bugs to do it.
Why do we add salt in this experiment?
That's a really good question. Well, I have some salt here, sodium chloride, very pure sodium chloride. Salt is used because when we're extracting DNA, salt is something that's going to take a lot of proteins away from the DNA. When DNA is in our body, it's wrapped up in something called a chromosome. Does that sound familiar? So a chromosome has a lot of proteins on it, and in order to get the DNA to be as pure as we can make it, we try and take all those proteins off so that we just get the DNA. So salt's really good at doing that.
And how about detergent?
Yeah, detergent, normal detergent can be used to purify DNA like we found out in lab. Detergent has something in it that we use every time we use detergent. It's a grease dissolver or a lipid dissolver. You know the word lipid?
Yeah.
Yeah, so lipid, right, like we learned about in class, is what exactly?
It's like a fat.
Yeah, like a fat or fatty acid. So that molecule gets broken down, just like when we're washing our dishes. When we use detergent, it breaks up grease. Well, the same thing happens with DNA. We want to break up the cells that have what's called a phospholipid bilayer around the cell. So it has fatty acids around the cell. We want to make sure that those are broken up so we can get to the DNA inside the cell. So there's something in detergent called sodium lauryl sulfate or sodium laureth sulfate, and that's very similar to what's used in the pharmaceutical industry to break up lipids and extract DNA.
How long will my DNA last? Like, will it disappear or will it stay?
In your body or outside?
Like the
when
one
you
I
extract
extracted,
it?
yeah.
Okay. So
like the
when you
one I
extract
extracted,
it.
yeah.
Okay. So hopefully it lasts a long time in your body, right? So when you extract DNA, you can actually keep it for pretty long amounts of time. People in the industry will recommend that eventually, within like a year or so, you may want to switch over to new samples of DNA just to make sure you have the freshest possible DNA. But DNA can be stored at minus 80 degrees Celsius, that's very cold, for a very long time. And it can be stored for, say, several weeks, maybe up to a month, at negative 40 degrees Celsius. And if you think you're going to keep it around for like a week or so, you can store it at about 4 degrees Celsius. So that's just above freezing.
Mhm. Can you extract human DNA using this protocol?
You absolutely can. Just like if we were to swab our cheek cells, you can actually use Gatorade, the everyday drink Gatorade, to perform a very similar experiment because of the amount of salts and electrolytes in Gatorade. Yeah.
Cool.
Yeah.
How is DNA extraction useful for scientists? Like, when they use this protocol, why is it important?
Well, it depends on what kind of science you're doing. So we learned in class about forensic science and how DNA can be used to extract, how DNA can be extracted and used to determine who was at a crime scene. It can also be used, like when I was in college and I worked with those bugs that I ground up, it can be used to figure out how different animals are related to each other based on how closely their genes match up. So that's really important. And it can also be used in studies of family history. So if people want to see what kinds of diseases are in their family, their DNA can be extracted, and very simply a look at their entire genome can be, or pieces of their genome can be determined. There are lots of uses for DNA extraction.
What do you think of DNA extraction?
I think it's really useful. I think there are lots of applications for DNA extraction. It's a fun lab to do. It's something we can do even in our kitchens, and it's something that I think there will only be further advances in it and in the ways we can use it. So it's a really exciting time to be a young scientist.
Let's move on to DNA extraction. How long have you been a teacher?
Well, this is my second year teaching full-time in a public school. I also taught in museums for about 8 years.
What's it like being a teacher?
Well, it's very busy. I definitely have worked on my people skills over the years and being able to work with students of all age ranges. And I think one of the best parts of being a teacher is being able to meet and interact with so many students and help them figure out what they want to do.
Why did you choose biomedical?
Well, I've had some experience in the biomedical field, and it was a really great opportunity to learn how to do this Project Lead the Way course. And I also teach anatomy, so it kind of tied in really well to my real passion for how the human body works and how animal bodies work, and my past studies in paleontology when I studied dinosaurs.
What do you do like in your free time?
Oh, my free time, I read a lot. I write science fiction, because I'm a nerd, and but proud of it. And I watch movies, hang out with my friends and my husband, and I now have a cat, a pet cat, so I'm very happy to have a little fluffy friend at home. So I also rock climb. I'm a rock climber, indoor rock climbing.
Wow.
Yeah.
That was actually my last
Yeah.
my next question. Do you have any pets? Do you have a cat?
I have one cat that we just got, and his name is Fievel. Yeah.
Do you like to read books?
I do. Mostly science fiction, fantasy, young adult literature. I also like to read nonfiction from time to time, science-related stuff.
What's your favorite book?
My favorite book ever? That's like asking me to pick my favorite student. I can't do that. If I had to choose, I'd probably say I really like Lord of the Rings. It's my favorite.
Yeah.
Yeah.
You like to watch movies?
I do.
What's your favorite?
Which do you think is my favorite?
Lord of the Rings.
Well, I like Lord of the Rings, but actually my favorite is probably Jurassic Park.
Oh,
I
yeah.
love Jurassic Park. It's good. Yeah.
What are your plans for the future?
Plans for the future? Well, I want to keep on helping students as much as I can for as long as I can. I enjoy working with teaching here. I enjoy working in museums, and I think no matter where I end up, I want to keep hopefully helping students. Like I had great great teachers when I was in school to help me figure out what I wanted to do in my life, and we're all still trying to figure that out. So yeah.
Let's play a little game. Basically the rule is to say the first thing that comes in your mind when I
Okay.
say this word.
Okay.
Teacher.
Work.
School.
Fun.
DNA.
Bugs.
Books.
Lord of the Rings.
Doctor Who.
Ooh, science fiction.
Harmony.
Community.
Students.
Students, I would say diverse.
Well, that's all. My last question is a multiple choice question.
Oh boy, okay.
The question is, do you like Harry Potter? And the answer choices are A, yes, B, A, C, B, and D, all of the above.
I would say D, all of the above.
That's the right answer.
Okay, good. I see you're taking a page out of my book, maybe. Do you like dogs? The answer is yes. That's an inside joke from our class. Okay.
Well, thank you for your time.
You're very welcome, Aisha. Thank you. All right. Bye.